Okay, so let's go back to our two-way ANOVA. Let's go to, so I want to get rid of that filter. So let's see, we filtered rows, so clear row filters. And you will see how well you did. If you do it, you should have 35,556, right? Okay. Now remember, we mentioned that you can have a lot of false positives. You're doing a lot of t-tests or ANOVAs. Here is what we can do to basically compute false discovery rates. So a p-value gives a probability of you arriving at that hypothesis by random chance for just that gene. A false discovery rate basically rates entire gene lists. So if I have a gene list of 100 and I have a false discovery rate of 10, I only expect 10 of those 100 genes to be false positives. We can do that. So go to stats, go to multiple tests, and go to false discovery rate. Do you have anything up here? You might have to fill some things in. So what you can do is I put 0.001, put a space, and then go 0.005, space 0.01. You can go up as far as you want. You just put a few in, 0.01, 0.05, 0.10. Now hit OK. I hope it doesn't bring my browser up. Oh, no it didn't. New window. This is basically telling me that at a false discovery rate, let's go to 1%. So you can see here, we could do the days, right? So at day, a p-value of 0.003 is going to give me a false discovery rate of 1%. So out of these 11,000, basically 11,400 genes, I only expect 1% to be false. That's a lot of genes that are changing that are significant. But you can see the other ones are very manageable. Cell type, about 372. Day and cell type, about 41, 282, etc. A lot of times, sometimes I'll do this and I won't get any genes that are significant at a false discovery rate. And most people will actually, I've seen bioinformaticians actually, they'll start taking data out so they actually get significance. Like they'll take samples out, they'll do the PCA and then they'll pull the samples out that aren't like, that don't separate, and then they'll do the test again so they can get it. And that is completely and utterly wrong. It kills me every time I see that too. So if somebody asked you, hey, how many genes did you get due to cell type at a false discovery rate of 1%, you would say, I got 372 genes, which means that 3.7, about 4 of these genes are probably false positives. This false discovery rate corresponds to a p-value of 0.000104624. Does anybody have any questions on this? Does everybody understand that? False discovery rates versus p-values. P-value is per gene, false discovery rates per list. The way we get that is we calculate, basically what we know is that, at this p-value, I only expect 35,000, take 35,000 times this, and I bet it's 1% of this. Did that make sense? That's exactly how they do this. That's how you calculate false discovery rates. So what happens if, and that happens too, right, is that you'll get nothing in the false discovery rate. And, I'll try to explain to researchers, and that's what they hate hearing is, they spent, tens of thousands of dollars on this experiment. You look at the data and you go, I'm sorry, there ain't nothing different. And then they look at you like, can you find something different? And I say, nope. Not if I make stuff, not unless I make stuff up, which I do not. But again, these are all just kind of tests to make sure, just give you an idea of the statistical tests and what kind of list you expect. That right now I know if I'm going to look at day, there's just almost everything's changing. But now when I look at differences in cell type and maybe day versus cell type, that's a little more manageable. Okay, so we can close out of this. So let's make some gene lists.